Hi all, welcome back. Before we start talking about disorders and diseases that are related to the nervous system, I want to spend some more time talking about action potentials, mainly because I think uh, when we covered it for skeletal muscles and how skeletal muscles receive a signal from a nerve, we didn't really talk about the cell potential. We didn't talk about action potential waves or anything like that. So I want to cover that material before we get into kind of disease disorders, that kind of stuff, mainly because a lot of those diseases, disorders, genetic issues play an important role in kind of action potential. Okay, so first off, cell membranes, they have a certain potential. Potential just means that it has a certain charge to it. Okay, so they're electrically charged or polarized. Okay, and the potential is, the difference that you can get between the intracellular fluid and the extracellular fluid. So there's a membrane here, which we already talked about a long time ago. It's phospholipid membrane. Okay, so it's made of fats and proteins, which doesn't allow charged molecules to move across. You have some charged molecules on the inside, you have some charged molecules on the outside. Okay? The polarity of this is the charge difference between the two. Okay? The potential, or the membrane potential, is the charge inside the cell. Okay? And for the most part, the inside of the cell is going to be negative respect in respect to the outside of the cell. Okay? And so we have membranes that have a threshold where they are balanced at a certain charge, a certain amount of millivolts, and we'll get into this in a little bit. When you have a imbalance of ions on one side versus the other, you can change the charge. And because you can shift material, you can shift the number of positive ions moving out and the number of positive ions moving in, you can change the charge over here. So if you shift more sodium out, less potassium in, right, this will become more negative and this will become more positive. Okay, and that's really how a nerve impulse works. It's the movement of ions across the membrane. It's active movement, meaning that we have to use energy to do this, ATP, but it's the movement of ions across a membrane, changing the membrane's potential, changing the polarity or the polarized sections of the material, which can cause a wave of energy to move across the membrane, okay, or move throughout a membrane. Okay? And so really it's a compilation of charge difference, okay, and the ability for cells to do this rapidly. Okay, so open ion gates, move ions across, changing the polarity on the one side versus the other. Okay, and that allows for communication between cells to occur. Okay? And we'll look at this from an action potential point of view next. Okay? All right, so membrane potential, the charge inside. Okay? The potential to transport charge across a membrane. That's what it really means. The resting membrane potential, okay, is about negative 70 millivolts in humans. Okay, so this is about where it is at normal status or inactive status, negative 70 millivolts. Okay. So that's the potential of movement across. So if we look at kind of this action potential, if we're sitting at -70 inside the cell, okay, and then we have a stimulus, and that stimulus allows for the ions, the ion gates in the system to change, and we become more, or we become less negative, so more positive, okay, there will be a threshold. There will be a point at which, okay, depolarization will occur. Then you have this huge influx, okay, of material, either negative ions moving out, okay, or a massive number of positive ions move in. That's depolarization. So you go from negative charge inside to a positive charge, okay, and that carries a signal. Then, okay, those gates open and you get repolarization. You get negatives being pushed back in, positive pushed out, okay, and you go back to negative. Now you can have a refractory period, and we'll talk about this when you go too negative, too far negative, but it will go back to its resting state at -70. Okay, what's moving? Typically potassium ions and sodium ions. Okay, and so these are the ions that are typically moving. Okay, they're both positive charged, but you'll have other ions in the system. You'll have negative charged ions like chloride ions. You'll also have other things like proteins, which will have negative charge, which is building up part of this negative charge that occurs inside is the proteins that are within the cell. Okay, so again, what I just stated. So negative inside is due to the negative charge of proteins and ions like phosphate and sulfate and chloride. Okay, the movement of these ions across is what we call gated. It means that you have to use energy in order for the ions to move. Ions can't move across the membrane without a protein being in the membrane, a channel allowing for that to happen. Okay, and most of those channels, okay, especially in this case, especially when you're going against the gradient, if you're becoming more negative, okay, you're going against a gradient. If you're becoming more positive, you're going against a gradient. Many of those need energy to open those gates, open those channels. All right, so if we're looking at the potential and the distribution, again, sodium and potassium ions are going to follow diffusion rules. We already know how diffusion works, right? You go from high concentration to low concentration. Right, so if there's more sodium outside the cell, then the sodium's going to flow into the cell. If there's more potassium in the cell, the potassium's going to flow out of the cell. Okay? So that is diffusion. Okay? Sodium potassium pumps are active pumps that pump the ion across the membrane. This requires energy, requires ATP, and it can move ions from low concentration to high concentration against the rules of diffusion. Okay? So when we talk about kind of this resting potential, we're talking about 3 sodium ions being pumped out of the cell and 2 potassium ions being pumped into the cell. That's why the cell itself is negative. Okay, along with the proteins that are negative in there, okay, the chloride ions that are negative in there, etc. Okay? So you have to use ATP to move these things in the opposite direction. We already talked about that. Okay? That's where you have this normal status, is the sodium potassium pump is what generates this normal status of -70 millivolts. Okay, and that's what really, when it goes back to -70 after an action potential happens, okay, that's the resting phase. Okay, then it's ready for a new impulse, a new action potential. So it has to return back to the resting potential in order for it to start a new action potential. So this is how an action potential works. First of all, you have something that excites the cell, okay? An electrical event that can excite the cell. Okay? This is signal, this is a stimulus, this is really anything. That changes the membrane potential. It changes the charge inside, outside. Okay? First positive, then negative. Okay? Then it returns back to the potential. Okay? And then it's ready for another communication. Okay? So let's look at this. Okay? So here are those gated ions. Okay? You need energy to open these gates and allow for the ions to move across. It's just a protein. Okay, remember, the membrane itself doesn't allow ions to move across. The protein has to do that. So you have to have a channel. Okay? In this case, these channels are normally controlled by ATP. So when there's energy in the system, these channels will open and things can move back and forth. In some cases, they're pumped when the concentration, it goes against the concentration gradient. Okay, so if we're looking at a neuron and we have high sodium on the outside and low sodium on the inside, the sodium will move in. Okay, that's diffusion. If you have high potassium on the inside and low potassium on the outside, the potassium will move out. Okay, this is diffusion. You don't really need energy for this to occur. Okay. What happens is when the opposite is true. When this becomes high and you need it high sodium and you need it to be more sodium, then you need a pump to pump it across. Okay, and we've talked about this before. I just want to make sure that it's fairly clear. Okay, so here is that pumping action. Okay, so you have high sodium, low sodium. But then if you want the sodium out of the cell, which is what we do to stabilize it at -70, we take the sodium that's in here and we pump it out. Okay? And so you're pumping more sodium out of the cell than sodium that's coming in, okay? Which results in a negative charge. Okay? Same thing with potassium. Okay? Your potassium's moving out, but you're pumping potassium in. Okay, this creates a stabilized structure. Okay, but you can see you're pumping more sodium out than potassium in, okay, which is why it can maintain that negative status. Okay? All right, so let's look at an action potential. So the resting potential or the resting phase is -70 millivolts. Okay? For most well, for humans in general, the threshold is about -55. Okay. Then once you've pumped enough sodium, okay, and potassium in different directions, and you've pumped it enough to change the charge within the cell, okay, to get over that -55, that threshold, okay, then you get a depolarization. It means now you've created this flow of ions to the point where it's going from negative to positive. Okay, and when it does that, it allows for energy to flow through the system, through the cell. Okay, then you get a repolarization event. Okay, that if it goes past the threshold potential, which it often does, it will be called hyperpolarization. So I'm sorry, not past the threshold, but past the normal uh, status or the resting status, okay, of -70 or whatever it might be for different organisms, okay, you get this hyperpolarization. So you dip too low, okay, and then it rights itself. It goes through homeostasis. It rights itself to the resting potential until another signal stimulus is sent. So in order for this to occur, you have to have some kind of stimulus. And if that stimulus is not enough, and the stimulus is not great enough stimulus, okay, and it doesn't reach that threshold, it'll be a failed stimuli or a failed nerve impulse, and nothing will happen. Okay, and we'll talk about that here in a little bit. Okay, so they're excited by a response to some kind of stimuli. Okay? That stimuli is going to change the negative 70 millivolts in either direction. Okay? It could be positive, more negative. Okay? Neurons, which we've already talked about. Okay? You have neurons that have dendritic ends, axon ends. They're going to pick up some stimuli. The stimuli could be touch, it could be sight, it could be taste, hearing, smell, okay? It could be temperature. All these things will be some type of stimuli. Okay? That signal is sent as long as it meets the threshold. Okay? So when that stimulus happens, it opens up sodium channels and sodium enters the cell. Okay? That changes the membrane potential. Okay? And often we call membrane potential changes graded. Okay? Which means that you can have multiple stimuli needed in order for an action potential to occur. Okay? Or a greater stimuli. And we can change how much stimulus is needed for that action potential to occur. Okay? So if you overstimulate a region, okay, or if you continuously stimulate the same nerve over and over and over again, you could change how much stimuli is needed in order for that potential to change. Okay? So if you don't meet the threshold, no action potential occurs. Okay? Graded stimuli, again, can be added together, okay, to produce the threshold stimulus. So you might have multiple stimuli that are being sent to get one reaction. Okay, so you might get stimuli from the eyes, okay, from vision, smell, hearing, taste, touch, all of them getting a similar reaction to meet a certain threshold, okay, in a certain region. Okay, again, just to recap what we said before, threshold potential typically right around negative 55 millivolts. Okay, you need some kind of stimulus, okay, to reach that. Okay? It could be coupled. It could be a series of stimuli, okay, that allow you to reach that. It could be one strong stimulus that allows you to reach that. Okay, when that happens, the sodium channels are open. Okay, that is what we call the trigger zone. Okay, when you meet that threshold potential, it opens the voltage gated sodium channels, and now you're in trigger zone. Okay, trigger zone means that that action potential is moving. That's the action potential. It's moving. That signal is being sent. Okay, that wave of energy is moving across that cell. Depolarization, okay, is the change from negative to positive charge inside the cell. Okay, making both sides positive. Okay. When this occurs, the gates will close. Okay. This is what we call the all or none law. Okay, so either you reach an action potential or you don't. Okay, there's nothing in between. Okay? And that means that when the signal is sent down the axon, okay, it's sent all the way down the axon. Unless there's damage in the axon, which we talked about with MS, okay, then the signal can't make it, okay? But if the action potential is sent from the neuron, it moves all the way down the axon. Okay, repolarization, this occurs after depolarization, okay? And now you have potassium channels open, and so the potassium channels can rush out and the polarity will return, okay? So sodium first, lots of sodium, changes it to a positive charge inside and outside, okay? Then repolarization, all the potassium starts to leave the cell, okay? Changes it back to a negative charge. Okay? Hyperpolarization is when you overshoot during that repolarization. We already talked about this. So you drop below -70, okay? And the sodium potassium pumps will return to the original concentration. Okay? So at any time, sodium potassium pumps along with ATP can be used to try to balance the concentration and return to that homeostatic stasis, which is going to be that -70 millivolts. All right, let's look at it. Okay, so you have some type of signal, in this case, a neurotransmitter coming from another neuron to this neural body. Okay, that stimulates sodium channels to be opened. Okay, and so there's receptors on this neuron that are going to open up these sodium channels. When that does, sodium starts leaking in to the cell body, and you start getting a cell body that becomes more positive. Okay? If it's only -62 millivolts, no signal sent. Okay? But if that neurotransmitter, if you get all these neurons coming in, okay, multiple neurons coming in and stimulating this, okay, multiple neurotransmitters, and you got all these protein channels now open, okay, so you get even more sodium flush in, okay, you reach your threshold, then you have that depolarization where now the inside the body becomes positive along with outside the body, okay, and you get a signal, a voltage signal being sent down the axon. Okay, so movement of the ions during the action potential. Again, sodium's coming in during the depolarization. You get this depolarization, sodium's in, potassium channels are closed. Okay, and then the repolarization, potassium channels are open, sodium channels are closed. Okay, and you can repolarize. Okay, again, it's for most students it's not intuitive because both molecules are positive, but remember, okay, more sodium molecules are moving in than potassium molecules are moving out. Okay, depolarization, it's becoming positive, then those potassium molecules start moving out, okay, to correct for the sodium molecules that moved in. Okay, and you get repolarization, it going back to negative. Okay, and then of course, if you overshoot, you get the potassium, sodium potassium pumps pumping things back in to kind of restabilize it. All right, so what's leading up to this? Again, just to go through the steps of an impulse, just so you know how your nerves work. Okay? First, your cells are going to be at their resting potential. Okay? The nerve cell itself is going to be at the resting potential. Sodium and potassium pumps will even out and make sure that it's at that resting potential. Okay? That neuron's going to receive some stimuli. It's going to receive a stimulus, whatever that might be. Okay? Again, taste, vision, touch, okay, temperature, anything that can produce a stimulus, which is going to send neurotransmitters to a different neuron. Okay? So that stimulus occurs. The sodium channels will open. okay, and move into what we call the trigger zone, okay, which means that they're all going to be open and that body is going to become positive. Okay, so sodium ions diffuse inward, depolarizing the membrane. Okay. Then, after that has occurred, you'll have a repolarization phase. Potassium channels open, potassium moves out, repolarizing. Okay. If it goes too far, it'll be hyperpolarized, okay, and you'll need to correct for that. But in this action of depolarization, repolarization, an action potential moves, okay, and it moves down the axon, okay, so it goes all the way, the whole length of the axon. All right, let's look at the refractory period versus the relative refractory period versus the absolute refractory period. Okay, so during these impulses, you have really two parts. The absolute, okay, this is where sodium and potassium are moving because the gates are open. Sodium first, potassium next. Okay. And then you have a relative refractory period. Okay. And this is where you're starting to move into the resting phase. Okay, so you can't have another action potential occur until you've reached the resting membrane potential. Okay, so you must reestablish resting potential. Okay, otherwise, depolarization can occur again. Okay, and so we often will talk about refractory periods, and they limit how often an action potential can be sent. Okay? So it limits the number of action potentials that can be sent in a given period of time. Okay? And that really has to do with open channels, closed channels, and then establishing that normal resting state. Okay? All right. Again, back to the nerve itself. Okay? The speed of the impulse, okay, varies with myelination. Okay? So unmyelinated axons conduct impulses over the entire length. Remember, there's no regions where sodium can't penetrate. Okay? So they can have a signal go across the entire length. When we talk about myelinated axons, myelin is rich in lipids, and we already talked about this. Okay, so it doesn't allow ions to move in to the system. Okay, and ions will only move in the nodes of Ranvier. Okay, we already kind of touched upon this. Okay, so myelinated axons, okay, will transmit an impulse through saltatory conduction, okay, in which the action will jump from one node to the next node. Okay? And so I'll show you a clip of this, but only at the nodes of Ranvier can you have sodium enter. So the action potential has to jump, it has to move along those nodes. Okay, but that really allows for the faster impulse than an unmyelinated axon. So unmyelinated axon, you have sodium channels opening all along there, all along the entire axon. Okay, when you have them only opening at nodes of Ranvier, okay, then the action potential jumps from node to node over the myelinated sheaths, okay, regions, and it's jumping, okay, and it's faster. The other thing is axon diameter will affect how fast an impulse moves. So a thicker axon will transmit faster than a thinner axon. Okay, and so thicker are myelinated and thinner are unmyelinated, and it's significantly faster to have a myelinated axon than an unmyelinated axon. Okay, 120 meters per second, okay, versus zero point 0.5 meters per second. Okay? So let's look at this just to make sure that you understand what's going on. So again, we have this action potential. We have this sodium traveling in, okay, potassium traveling out during the repolarization phase. So depolarization, sodium's traveling in. That starts the action potential. But instead of having action potential all along, okay, it jumps. Okay, so you have a refractory period and action potential, ready for an action potential. So it jumps between the nodes. Okay, and so each region is ready for a reaction after that occurs. So this region would be in refractory period. This one would have an action potential. This one would be ready for the action potential to travel down it. Okay. This one over here will be ready for another signal to send. If it's unmyelinated, that's not the case. Okay. So it's less compartmented and it's more a unison across the entire thing. Okay, and so that's what slows down the signal. When it can jump from nodes of Ranvier, okay, it moves faster down the sheath or down the axon. Okay, so factors that might impact the impulse conduction, okay, is how permeable the axons are to certain ions. Okay, and we already kind of talked about this. Remember, that if you have a bunch of myelation on the sheath itself, okay, that decreases the permeability. Okay? If you have lots of sodium and lots of potassium inside extracellular fluid, okay, then you can change the potential. You can change the speed at which a signal is sent. Okay, so how much potassium is in the system, okay, will tell you basically the threshold that you have to reach from a stimulus to change or to generate an action potential. Okay, this is why you have things like cramps and other things that are occurring in your body is often the result of potassium and changes in potassium. Okay? When we have a lack of potassium in the system, when you go through hyperpolarization, okay, your action potentials tend to not be generated. Okay, so they're hyperpolarized, so action potentials can't be generated. Okay, which will lead to a muscle being going through paralysis or not being able to be stimulated. Okay, so you have two systems here. If you have too much potassium in the system, you can have your muscles kind of stimulate too much, okay? And you can have your muscles firing through what we call convulsions, okay? And so uncontrolled, and you'll often have your muscles just firing over and over and over again, okay? And that's because you have too much potassium in the system. If you don't have enough, okay, it goes through paralysis and the muscle can't fire. Or maybe it maintains tautness and doesn't release. Okay. And then the last one is you can alter, okay, the permeability of ions by taking some things like different types of drugs. So anesthesia is doing this when you have an anesthetic drug that's put in your body. It changes the permeability of potassium ions, okay, which will change the amount of ions that are around in order for an action potential to occur. And normally what occurs is you get paralysis, you get a lack of impulse, a lack of um, a lack of pain receptors is a good example of this. This is why you would use an anesthetic. Okay, you're trying to deaden the pain so the individual can't feel the pain. Okay, and it's not changing anything except for the permeability of potassium ions. Okay, and we'll come back and we'll talk about other ways in which this can happen naturally, where you can have issues with potassium or sodium in the system.